Hi dear friends, today we are going to discuss about micropropagation principle, stages and application. We all know what is micropropagation. Micropropagation is the artificial process of producing plants vegetatively through tissue culture or cell culture techniques. Cornell University botanist Frederick Campion Stewart discovered and pioneered micropropagation and plant tissue culture in the late 1950s and early 1960s. In this artificial process of propagation, plants are produced in vitro by asexual means of reproduction or by vegetative propagation. In this technique, where a callus mass has been initiated from a single explant taken from any living part of a donor plant, and within very short time and space, a large number of plantlets can be produced. Plants can be produced both asexually, that is via vegetative parts multiplication, or sexually, that is seed production. One of the means of asexual reproduction is by multiplying the genetic replicas of plants that are referred to as clonal propagation, wherein plants can be propagated from a single individual through sexual means of reproduction. It is also used to provide a sufficient number of plantlets for planting from a stock plant which does not produce seeds or does not respond well to vegetative reproduction. Let me discuss about the stages of micropropagation. There are mainly 5 stages. The stage 0 is the selection of an explant. Stage 1 is the culture initiation and establishment. Stage 2 is shoot multiplication. Stage 3 is rooting of the shoots. And stage 4, transfer of the plantlets in the greenhouse environment. Stage 0. This is the initial stage of the micropropagation. The stock plants are selected and grown under the controlled conditions before using them for culture initiation. Stage 1. The explants are established in a suitable culture medium. These stages involve the following steps, that is the isolation of the explant, surface sterilization, washing. The surface sterilization we mainly do by the distilled water, mercury chloride, alcohol, 220, etc. The explant is established on an appropriate culture medium. The stage 2 is in this stage involves the rapid multiplication of the shoots or rapid somatic embryo formation in a defined culture media. Stage 3. In this stage, the shoots are transferred to a medium for the development of roots. The shoots are either transferred directly in the soil for the root development or transferred to a nutrient medium in the development. The stage 4. In this stage, the plantlets are established in the soil. The shoots from the laboratory are transferred to a greenhouse under controlled conditions of the temperature, humidity and light. This is the picture showing the micropropagation steps. The stage 1, establishment of this is the stage 0, that is explant source. We take this selection of the explant. The stage 1, the establishment of the explants in the culture. Shoots are established in the culture 2 to 3 months. Development of the small clusters of the shoot. Stage 2 is the shoot multiplication. We can see the small clumps of the shoot increased. Next is the cycle 3, the normal the increase of the shoot length. Stage 3, in vitro rooted in the culture for 1 month, that is rooting, or ex in vitro in the direct soil. The plant established in individual cells 3 to 4 weeks under mist. Total growth period in cell is 3 months. Next, the 4th stage is the acclimatization. That is, it can be transferred to cans or the sailing covers for the sale as a saleable plant. Next is the methods of micropropagation. This artificial process of producing plantlet involved mainly 5 different methods. First is the meristem culture. In this method of propagation, subtending leaf, primordial and a meristem is placed into their respective growing media culture and allowed to grow. After some weeks, an elongated rooted plant is produced. Once after they reach a considerable height, these plantlets are transferred into the soil. In this method, a disease-free plant can be produced and can be successfully used for rapid multiplication of various herbaceous plants. Second is the callus culture. In this method, selected plant tissue is placed in an artificial growing medium culture until the callus is formed. After the production of callus, they are transferred into a culture medium containing plant growth regulators for the induction of adventitious organs. After a few weeks, a new plantlet is exposed gradually to the environmental condition. The third is the suspension culture. In this method of micropropagation, cells or group of cells are dispersed and allowed to grow in an aerated and sterile liquid culture medium. The fourth is the embryo culture. In this method of embryo culture, the embryo is extracted and placed into a culture medium with proper nutrient in aseptic condition. The fifth is the protoplast culture. In this method, the plant cell is isolated and cultured in an appropriate medium to reform the cell wall and callus. Later, under suitable conditions, the cell develops a cell wall followed by an increase in cell division and cellular differentiation and grows into a new plant. Next is the application of micropropagation. This is an alternative method for vegetative propagation with enhanced multiplication rate. Large quantities of identical plants can be obtained from a single plant tissue within a very short time period. The small-sized propagules can be stored and transported easily. It helps in the production and maintenance of pathogen-free plant varieties. It is a cost-effective process. New varieties or species can be propagated. A requirement of less space and human resources. This method is independent of season and can be carried out anytime. Millions of plantlets can be maintained in the culture vials. The germplasm stocks can be maintained for several years using this technique. Genetic uniformity of the propagules can be maintained through this technique.